All right, last topic for chapter 6 now is going to be binary fission. With binary fission, what we're really talking about here is going to be cell division. What that cell division is going to be an asexual form of reproduction. All right, the green pen's squeaky, so I'm going to switch to blue. That asexual reproduction, all right, essentially what you're generating is going to be an exact copy. All right, so you're generating a clone here with the bacteria. All right, and in terms of that binary fission, what you're going to have happen, if I draw a generic bacteria up here, this is our DNA. I'll draw the cell wall. Inside's the cell membrane. All right. What ends up happening in binary fission, the DNA is going to replicate. And when we get to the unit on microbial genetics, that's where we're going to discuss the nitty-gritty details of DNA replication. Right now, I want you to just know the DNA replicates, so you end up with a copy. The cell will also elongate. All right, and I'm going to draw the one DNA here in blue, just for fun, so you know this is the copy. That one's going to be in pink. Okay, so you've got the DNA that replicates. And then the cell's going to split. This is the part that makes up the binary fission. Fission meaning dividing something essentially into parts. So we're going to divide that cell into two parts. I'm going to try and draw that as best I can. All right. Essentially what you've got the cell wall there. We've got our DNA from our one cell. We've got our exact copy that's in pink in the other cell. This is the fission portion. You're dividing that cell wall and that cell membrane. Eventually what's going to happen, that's going to pinch apart. And we're going to have two genetically identical cells. Okay? So in terms of bacteria, when we talk about binary fission, very often what you will hear is that expressed in as a doubling time. All right? So doubling, going from 1 to 2. Okay? And in terms of that, most bacteria have a doubling time between roughly 20 minutes and several hours. Again, it depends on the type of bacteria. This is also where with binary fission those environmental factors come into play, because if you aren't at an optimal temperature, it's going to take longer for that bacteria to replicate its DNA and make a copy. All right? If you're at the ideal temperature, it might be as short as 20 minutes for that bacteria to undergo that whole process of binary fission. Okay? So the doubling time is what we refer to with bacteria, and that occurs logarithmically. Okay? So in terms of that, 1 goes to 2. Those two cells now can divide, and we've got 4. All right? Those 4 divide, we've got 8, so on and so on. I'm not going to ask you to do the math in any of the quizzes or the tests, but that's how bacteria can really start to take off in a particular environment. Like the example I already gave with growing bacteria in the potato salad. When that potato salad sits out, you may start with maybe, say 5,000 bacteria in that whole potato salad mix that you've got. And at that level, that number of bacteria isn't going to make someone sick if they consume it. But if we go 20 minutes, sitting out, nice warm temperature, those bacteria, those 5,000 bacteria become 10,000 bacteria. Those 10,000 bacteria become 20,000 bacteria. So you can see how that can escalate very quickly and why you've got to limit exposure of certain foods so that they don't spoil, so they don't go bad. Okay? In terms of that doubling time, that's how we refer to that. And in terms of your PowerPoint slides, I think I have some examples of some times in there for some different bacteria. Those would be the only ones that I would have you accountable for on a quiz or a test, are the ones in the PowerPoint. All right, there's variation depending on the type of bacteria. Okay, and that's really why I put the slide in there, because some are shorter, some are longer. All right, and in terms of readout for bacteria, the very end of the PowerPoint slides, I've got some different methods for determining how many bacteria you have in a particular sample. Those are what I want you to know are ways to determine essentially bacterial growth. Okay? In terms of that, we've got the spectrophotometer, that essentially what it's looking for is cloudiness. The more cloudy that the media is, the more bacteria that you have. So it's not going to give you an exact cell count, but it's going to give you a rough estimate. All right? You also have in there, I think as an example that I have, is fact sorting or flow cytometry. That flow cytometry is a much more accurate readout on the number of cells that you have in a particular culture. Essentially what it is a fairly substantial piece of equipment. If you ever get into doing any research or go into a research lab, most major universities like University of New Mexico are going to have facilities where you can count the number of cells within a sample. And essentially what it is it's a laser, and it essentially reads the diffraction that happens when something passes through that laser. That individual cell passes through the laser, disrupts the laser beam. That ticks off the counter, and the fact sorter count the number of cells that you've got in a given sample. So this one's really accurate for determining the number of cells. All right. The other thing that you can use is cell cytometry. And with cell cytometry, you're actually counting the number of cells in a particular sample using a microscope. Okay? Really what I want you to know, those are the main pieces of equipment that can be used to determine cell number in a culture of bacteria. These methods can also be used with eukaryotic cells also. It's not exclusive just to bacterial cells, but they can be used to determine bacterial cell number. All right, so that's what I want you to keep in mind with that. This kind of wraps up Unit 6. Next, we've got Unit 7 or Chapter 7 coming up next week. Again, if you have questions, please let me know. Thank you for your attention.